Good morning, everyone. My name is Carley McIntosh, and last summer I had both the honor and the privilege to work under the mentorship of Dr. Sunday Charlotte and Dr. Marina Prepeliuk in the Department of Pharmaceutical Sciences at Thomas Jefferson University on a project entitled The Stable and Efficient Transfection of siRNA for Mutated KRAS Silencing Using Novel Hybrid Nanoparticles. So before I delve into the specifics of my research, allow me to provide you with some background information. Cancer is the name given to a range of over 100 diseases characterized by abnormal cell development. These cells have the ability to control their own cell division and infiltrate and destroy healthy body tissue. Cancer affects many parts of the body, including the lungs. Lung cancer is a strain of cancer which originates in the lungs. The lungs are a pair of spongy-filled organs located on each side of the thorax. Depicted in this picture, the lungs are connected to the trachea, more commonly known as the windpipe. As the trachea draws near the lungs, it diverges into two tubular branches known as the bronchi. And once the bronchi enter the lungs, they separate into several microscopic lanes known as bronchioles. The bronchioles end in alveoli, which are microscopic sacs of air. While the causes of some lung cancer cases are unknown, lung cancer often occurs when the cell lines of the bronchi and the parts of the lungs, such as the bronchioles and alveoli, are negatively affected by the chemicals they encounter. Lung cancer is split into two major types: non-small cell lung cancer, or NSCLC, and small cell lung cancer, or SCLC. Non-small cell lung cancer makes up the less aggressive form, which amounts to almost 90% of all lung cancer cases. Non-small cell lung cancer is the more common form because it has a wider variety of causes. And these causes are broken down into two major categories: inherited gene changes and acquired gene changes. While inherited gene changes are an identified cause of lung cancer, it's more often caused by acquired gene changes. Acquired gene changes are developed by one's exposure to certain chemicals or gases. They're known as acquired because they're brought on by one's own lifestyle or life choices. For example, those who choose to smoke are frequently exposed to over 100 carcinogens found in the tobacco and cigarettes. When one smokes a cigarette, any of these carcinogens have the ability to enter a lung cell and alter its DNA structure, leading to a cancerous mutation. In addition, radon, a radioactive gas, as well as asbestos and diesel exhaust, are also linked to the development of acquired gene changes. Several treatments have been created in order to repair the damage done by acquired gene changes, and of these treatments are targeted drug therapies. Targeted drug therapies target specific abnormalities within certain cell types and aim to fix these defects in order to prevent the spread and growth of one's cancer. RNA interference has become a highly investigated form of targeted drug therapies. siRNA is one of the most studied forms of RNA interference. This uses a synthetic double-stranded RNA molecule which silences the cancer gene through the destruction of mRNA molecules. While siRNA shows promise in its ability to efficiently silence the cancer gene, it lacks a successful delivery system. In order to create an efficient delivery system for siRNA, there are four key characteristics needed. First, the system should be able to protect siRNA from nuclease degradation during transfection. Secondly, the system should ensure minimal cell-mediated effect. The system should be able to escape endosomes after endocytosis. And lastly, it's important that the system does not elicit any inflammatory or immunological response. And after looking over these characteristics, the nanoparticle delivery system tested in this experiment was created. These nanoparticles consist of an siRNA model known as SIG12S, loaded inside a human immunoglobulin type IgG, coated with a poloxamer 188. SIG12S is an siRNA model created for the sole purpose of preventing the KRAS mutation found in A549 cells. This is an important testing subject because cells with this mutation have been proven to be resistant to chemotherapy treatment. And this SIG12S is loaded inside a human immunoglobulin. For this experiment, type IgG was used. IgG is one of the most common antibodies found within the blood. Therefore, by loading our siRNA in the IgG, it's disguised among the body's natural antibodies, making it less likely for the immune system to access or attack our SIG12S. And lastly, the IgG is coated with a poloxamer 188. Poloxamer 188 is a non-ionic carbon molecule which prevents the macrophagal intake of our nanoparticles. Each ingredient contributes to the safe and efficient delivery of the siRNA to the A549 cells. This leads me to my research design. The The objective of this experiment was to demonstrate the efficiency of the SIG12S in silencing the KRAS mutation when loaded inside our nanoparticle delivery system. It was hypothesized that the nanoparticles carrying the SIG12S would be more effective in treating the lung cancer than the chemotherapy. Please take a brief moment to look over the materials used, all of which are important to the success of my experiment. In order to test the nanoparticles' efficiency, we first needed to build the nanoparticles themselves. Once the nanoparticles were complete, they were ready for mass treatment. After treatment, the mice were sacrificed for lung extraction. And lastly, Western blot was performed to produce the results. To begin the formation of our nanoparticles, SIG12S, IgG, and poloxamer 188 were all dissolved in a low concentration of hydrochloric acid. Consequently, they acquired a pH of around 2. While the acidity created an ideal environment for the nanoparticle ingredients to dissolve in, the pH needed to be raised for the actual formation of the nanoparticles. As a result, the solution was slowly titrated with sodium hydroxide, our base solution. During this process, several drops of NaOH were transferred to our nanoparticle solution until it rose back to the optimal pH of 7. Once this was reached, our nanoparticles were spontaneously precipitated. To rid the solution of any loose ingredients, the nanoparticle suspension was centrifuged at 2000 RPM for 5 minutes. After, the supernatant, which contained any extra or loose ingredients, was discarded. The newly formed particles were then snap frozen in liquid nitrogen and loaded into a freeze dryer where lyophilization was performed for the duration of 48 hours. After lyophilization, our nanoparticles were complete and ready for mass treatment. 20 immunodeficient female mice were evenly split into 4 treatment groups. These groups consisted of a chemotherapy treatment group, which received treatment every Monday through Friday by oral lavage, a nanoparticle treatment group, which received injections of our particles twice a week on Mondays and Fridays, a combination group, which received both the nanoparticle and chemotherapy treatment, and lastly, our control for the experiment, which received injections twice a week, which contained PBS. All treatments occurred for the duration of 4 weeks. After the treatment process was complete, it was time to extract the lungs and observe the nanoparticles' effect. To begin lung extraction, all mice were sacrificed by the method of euthanasia. The lungs were then extracted from each mouse. One lung was saved for histology, while the other was separated into 3 parts, one of which we saved for Western blot. This was immediately snap frozen in liquid nitrogen and stored at -80°C to prevent degradation. Western blot began with the process of gel electrophoresis. We started by assembling the MES buffer, which was used to fill both sides of our kit. Next, samples from each treatment group were loaded into the wells of the gel. We ran our gel for 1 hour and 10 minutes with a voltage of 200. Once gel electrophoresis was complete, blotting was able to begin. First, we prepared our transfer buffer. Once this was ready, we could begin assembling our gel membrane sandwich, and each part to the sandwich was soaked in transfer buffer before being added. This consisted of 2 blotting pads, followed by a filter paper, our gel from the previous gel electrophoresis procedure, a nitrocellulose membrane, another filter paper, and another 2 blotting pads. Our sandwich was entered to the kit, and the transfer was run for 2 hours with a voltage of 25. After, we removed our nitrocellulose membrane and incubated it with water for 10 minutes. It was then incubated for 30 minutes with blocking solution, and after, the blocking solution was discarded and replaced with transfer buffer. And this was left on the stir over the night. The next morning, the transfer buffer was discarded and replaced with primary antibody solution. We left this on the stir for 5 minutes. Once those 5 minutes were complete, the primary antibody solution was discarded and replaced with new solution. And we did this 3 times at 5-minute intervals. After the 3rd wash, the primary antibody solution was discarded and replaced with secondary antibody solution. And this was left on the stir for an hour. After, we washed our membrane again, however this time we used secondary antibody solution. After washing, we could process our membrane to produce the results. And this leads me to my results section. Before looking at the silencing of the KRAS protein, we looked at the overall lung cancer development within each treatment group. To do this, each mouse was injected with luminal substrate so the intensity of the mouse's cancer could be monitored. Colors such as red and orange represented a higher intensity in cancer, while colors such as purple and blue represented a lower intensity. We saw the lowest intensity within our nanoparticle treatment mouse, which showed almost no signs of high-intensity cancer throughout the treatment process. Next was our combination mouse, which began to show small signs of high-intensity cancer within the first week of treatment. Around week 3, we began to see signs of metastasis within the tail. Followed by the combination mouse was our chemotherapy mouse, and similar to the combination, it began showing small signs of high intensity in the first week. However, this mouse's cancer was deemed more intense because of the signs of metastasis at that time as well. And lastly, our combination mouse had the highest intensity cancer with the most signs of metastasis. We then quantified the information from this picture. The line marked by the right side up triangle represented the nanoparticle treatment mouse and showed that while all other mice experienced a rise in their cancer intensity throughout the 4-week process, the nanoparticle treatment mouse maintained a low intensity cancer. After looking at the overall lung cancer development, we looked specifically at the knocking down of the KRAS gene. The results from Western blot showed weaker signals from our nanoparticle and combination wells and stronger signals for our chemotherapy and PBS. Each signal was justified by beta-actin, our housekeeping gene, which showed consistently strong bands throughout the wells of the gel. This meant that any lack of signal from the nanoparticle or combination wells was due to the successful silencing of the KRAS gene, not a measurement error. And this leads me to my discussion. Before I conclude, allow me to restate my hypothesis that the nanoparticles carrying the SIG12S would be more efficient in treating lung cancer than the chemotherapy. This hypothesis was accepted. The stronger signal of the chemotherapy confirmed its inability to treat the KRAS mutation cancer, while the weaker signal from the nanoparticle well demonstrated the ability of the SIG12S to knock down the KRAS mutation when loaded inside our nanoparticle delivery system. For future research, I would like to test the nanoparticle delivery system loaded with other siRNA types or other drugs in general. In addition, I would like to compare the SIG12S efficiency to other lung cancer treatments. I'd like to acknowledge the following people, all of which were instrumental in the completion of my project. Thank you for listening, and I now open the floor to any questions, comments, or constructive criticism. Thank you.